Transplanting pig organs into human bodies sounds incredibly crazy to most people. However, at the end of October 2021, several foreign media outlets simultaneously reported on a unique clinical study being conducted at the Langone Medical Center of New York University in the United States. This study marks the first publicized surgery worldwide where genetically engineered pig kidneys were transplanted into humans. What makes this surgery particularly special is not only the transplantation of genetically engineered pig organs into human bodies, but also because the organ recipient was not a typical patient with kidney failure. Instead, she was a woman diagnosed with brain death but still exhibiting signs of life with the consent of her family. If the surgery is successful, it would mean that genetically engineered pig organs could be safely used to treat human diseases. This would undoubtedly be a significant breakthrough in addressing the shortage of organ donors for transplantation. Hello, everyone. I am a member of AI Health Center, one of those dedicated to disseminating medical science and global healthcare knowledge. In this episode, we'll discuss this cutting-edge medical advancement and see what breakthroughs scientists have achieved in turning the seemingly crazy into reality. Transplanting cells or organs from animals other than humans into the human body is known as xenotransplantation. In fact, hundreds of years ago, humans attempted xenotransplantation. In the 17th century, bloodletting was popular in Europe for treating various diseases. However, excessive bloodletting inevitably led to ischemia in the human body. At that time, King Louis XIV of France's personal physician, Denis, experimented with transfusing lamb or calf blood into humans through veins. Although these xenotransplantation attempts were among the earliest, they undoubtedly caused serious side effects and even death. Therefore, they were explicitly prohibited by the French government around 1670. It wasn't until the 20th century that humans discovered the differences between different blood types and established a theory of immune rejection among different species or even different individuals within the same species. Subsequent research on xenotransplantation entered the scientific track after the discovery of immunosuppressive drugs that could alleviate immune rejection reactions. In 1964, a professor at Duke University in the United States began attempting to transplant chimpanzee kidneys into humans. However, most patients survived for no more than 2 weeks, with only a few surviving for several months. Subsequently, researchers also attempted to transplant baboon kidneys and livers into humans. And there were intermittent attempts to transplant chimpanzee and baboon hearts into humans. Unfortunately, immune rejection reactions caused by xenotransplantation were far more severe than those of allogeneic transplantation. It was proven that relying solely on immunosuppressive agents made it difficult for these patients or transplanted organs to survive beyond a year. Therefore, for more than a decade after the 1960s, research on transplantation was almost stagnant. However, after the 1980s, the number of patients requiring organ transplantation therapy began to rise sharply, and the problem of the shortage of human donor organs has persisted to this day. This has led to a resurgence of research on xenotransplantation. And through continuous exploration, pigs have gradually established a central position in xenotransplantation. According to the World Health Organization and the International Xenotransplantation Association, pigs are currently the only available source of xenotransplantation donors. Some may wonder why pig organs are more suitable for transplantation into humans than those of primates such as monkeys and chimpanzees, which are genetically closer to humans. Firstly, although primates are genetically closer to humans, their sizes are significantly smaller, making their organ sizes and functions often inadequate for human needs. Secondly, the reproduction efficiency of primates is low, and they often give birth to only one offspring per litter, making it difficult to meet the large demand for human organ transplantation. Finally, precisely because they are genetically closer to humans, many viruses and bacteria that infect them can also infect humans, significantly increasing the risk of infection after transplantation. Pigs, on the other hand, have clear advantages in all 3 aspects compared to primates. Firstly, pig organs are more similar in size and function to those of humans. For example, the size of a pig heart is similar to that of a human heart. And the average heart rate per minute is 50 to 60, very close to that of humans. Secondly, pigs have high reproductive efficiency and can be selectively bred. Thirdly, viruses infecting pigs are relatively less likely to infect humans compared to those in primates. Therefore, internationally, pigs are unanimously considered the best choice for current xenotransplantation. In recent years, the emergence and development of genetic technology have given hope that these pigs can be improved through genetic modification to become even more perfect. In fact, as early as the beginning of 2020, the prestigious academic journal Science predicted that a human transplantation experiment would become one of the top 10 scientific headlines of 2020. Although it was slightly later than expected by about a year, this still indicates that humans have long been confident in solving the related issues of xenotransplantation. In pig cells, there is a gene called alpha-gal that researchers found encodes a protein that is most likely to cause immune rejection reactions in humans. In this kidney transplantation surgery, the pig used was not an ordinary pig, but a genetically engineered one in which the alpha-gal gene was knocked out at birth. This allows the organs of this genetically engineered pig to avoid attack by the human immune system after being transplanted into humans. In fact, scientists are continuously using genetic technology to transfer human genes into pigs, while knocking out pig genes that may cause immune rejection reactions or safety risks, thereby constructing and continuously optimizing genetically engineered pigs specifically for xenotransplantation into humans. Before conducting human trials, scientists have transplanted organs from genetically engineered pigs into chimpanzees, monkeys, and baboons, observing that the longest survival time after kidney transplantation exceeded 1 year and after liver transplantation exceeded 200 days. Of course, these genetically engineered pigs have been fundamentally different from ordinary pigs since birth. In addition to genetic modification, they have been artificially fed in a sterile and virus-free environment since birth. Therefore, their future prices will also be high. Fortunately, after hundreds of years of exploration, humans have finally moved away from the initial madness and gradually seen the dawn of transplantation supported by science and technology. It is hoped that in the near future, these organs will be able to save the lives of thousands of people. If you find this medical knowledge helpful and inspiring, please follow AI Health, like, and share. Thank you!